We have a few rapid talks from far and near to Vancouver, and I will introduce the first speaker, which I think we're going to be watching a video for the talk from Danielle Gonzalez Carter from Innovation Center of Nanomedicine in Kawasaki, Japan.
Good afternoon. Firstly, let me apologize for not being able to give my presentation live. Second of all, I would like to thank the organizers of the Vancouver Nanomedicine Day for this great opportunity to present the work that we are carrying out here at the Innovation Center of Nanomedicine in Japan, in affiliation with the University of Tokyo, to target nanoparticles to the brain by exploiting the blood-brain barrier impermeability to label the brain endothelium. So all current strategies to deliver nanoparticles to the brain rely on functionalizing nanoparticles with ligands which bind proteins expressed by brain endothelial cells. However, all of these strategies have inherent brain specificity limitations, as these proteins are also significantly expressed on peripheral organs. Therefore, there is a high rate of off-target nanoparticle uptake, which decreases therapeutic efficiency, increases detrimental side effects, and ultimately limits the clinical translation. Therefore, we have created a new brain targeting strategy, which actually exploits the blood-brain barrier impermeability itself to generate the brain-specific targets, thereby increasing brain specificity. So the general gist of the strategy is as follows. Firstly, we label the endothelial cells of peripheral organs and the brain with a molecular label. Then, because of the lower endocytic rate into brain endothelial cells compared to peripheral endothelial cells, which is a crucial characteristic of the high impermeability of the blood-brain barrier, this molecular label is endocytosed and therefore removed from the peripheral endothelial cells at a faster rate than brain endothelial cells, so that after a certain period of time, the molecular label is selectively displayed only on the brain endothelial cells. Then, by creating nanoparticles functionalized with a linker which binds efficiently to the molecular label, we should be able to have no peripheral targeting and only specific targeting of the nanoparticles into the brain. Now, we have demonstrated the feasibility of such an approach in a paper recently published in PNAS, where we used a biotinylated anti-PKM1 antibody to label endothelial cells, and polymeric nanoparticles functionalized with avidin proteins to act as the linker to bind the biotin targets. Further description of the nanoparticle synthesis and all in vitro characterization can be found in our poster. Therefore, if you have time, please visit poster number 47 for more information. Right now, though, I will only focus on the in vivo data. And as I say, we injected biotinylated anti-PKM1 antibodies intravenously into mice. Then after a time interval of 15 minutes, 2 hours, 4 hours, or 8 hours, we injected the avidin nanoparticles, and 16 hours later, we quantified the increase in nanoparticle uptake into organs. And so you can see that at the short time interval of 15 minutes, there is highest targeting of the nanoparticles to the lung, but there's also targeting to the brain, heart, and pancreas. However, as the time interval increases, the targeting to the lung, heart, and pancreas steadily decreases, while the targeting to the brain remains fairly constant. This means that at a time interval of 8 hours, the only organ that the nanoparticles were targeted to was the brain. And here we can see the increase in nanoparticle accumulation into fresh ex vivo organs by IVIS imaging and into fixed brain tissue slices. So I hope I have been able to convince you in this short time period that we have developed a new two-step brain targeting strategy which turns the problem of the high blood-brain barrier impermeability to our advantage to actually generate the required brain-specific targets for nanoparticle delivery, which should help improve therapeutic brain delivery by reducing off-target nanoparticle accumulation into peripheral organs, and also should allow us to expand the repertoire of candidate proteins which we can use for brain delivery by identifying proteins not only overexpressed on the brain vasculature, but also those ones which have differential endocytic rates. With that, I would like to thank all my colleagues at the Innovation Center of Nanomedicine, in particular Professor Kazunori Kataoka, all our funders and you for listening. And if you have any questions, please don't hesitate to send me an email. Thank you very much.
As he mentioned in his talk, you can email him for his questions as he's not available right now to answer them in person. The next speaker of this series is MariaPoley. She will be telling us about chemotherapeutic nanoparticles and female reproductive system. And she is from Technion in Israel. Please.
Hi, my name is Maria, and I'll talk about nanoparticle accumulation in the female reproductive system during ovulation and their effect on fertility and anti-cancer activity. Females are 51% of the total population, and nowadays the awareness for gender differences in medicine is increasing. The main physiological difference between men and women is the reproductive system. Females go through the menstrual cycle each month, it's cyclic, and it's subjected to female-specific hormonal cues. Here we can see the uterus and the ovary. Before ovulation, there's increased blood supply to supply nutrients and oxygen. New blood vessels are formed around follicles, and they are leaky due to high levels of VEGF. The abundance of leaky blood vessels in the ovary will cause increased accumulation of nanoparticles in the reproductive system during ovulation, during the estrous stage, compared to the non-ovulatory stage, the diestrous stage. This can have both positive and negative effects. If the nanoparticles hold a toxic payload, they can increase ovarian poisoning, causing infertility, but it can also be leveraged to treat reproductive tract diseases. How we tested it, so we intravenously injected 80 nanometer gadolinium-loaded liposomes to female mice at different cycle stages. And indeed, we saw that during the estrous stage, during the ovulation, 2-fold more liposomes reach the ovaries compared to the diestrous stage, and 2.5-fold more reach the uterus. But what happens during cancer? So both the tumor and the reproductive system grow new leaky blood vessels, so where the nanoparticles will go. We tested liposomal distribution in 2 cancer models. During breast cancer, ovulation overcame the tumor vascularization, and the nanoparticles were shifted towards the reproductive system instead of the tumor, decreasing treatment efficacy. But in ovarian cancer, the added vascularity of the ovarian tumor targeted the liposomes to the tumorous ovary and increased treatment efficacy. So what will happen to the ovarian toxicity when during the treatment of cancer? So doxorubicin-loaded liposomes are used in the clinic to treat female breast cancer patients. So we wanted to see what are the long-term consequences. So healthy female mice were treated 3 times with either free dox or dox-loaded liposomes and were allowed to mate. So although it took a long time, the group that was treated with free dox had successful birth rates with high viability of the pups compared to the untreated group. On the contrary, the dox-lipo treated group had decreased birth rate and a significantly decreased viability of the pups, which means that treatment with dox-loaded liposomes impairs fertility compared to the free drug. So we know that the menstrual cycle affects nanoparticle distribution, but does it translate to human, and how it should be considered in the clinic? Thank you for listening, and have a good night.
I'm not sure if she's present, but I have a question.
She is present.
I'm here.
Hi. I was wondering, since you did gadolinium and probably used MR to image for the distribution, what percentage of the particles were actually in the tumor? And
In the ovarian tumor?
yes.
So actually, we didn't image MRI for the ovarian tumor. It was quite difficult to do MRI imaging due to the small size of the ovary of the mouse. It was mostly at the uterus itself, so it was quite hard to quantify in the MRI. But according to the liposomal quantification in the tumor itself, it was a bit higher than the ovary itself, about 3% of the injected dose. Not a lot, but still significantly different than the healthy one.
Okay, thank you.
I might have a question. Are there any other diseases other than cancer that might be, benefit from your approach?
Actually, I think so, because in a polycystic ovary syndrome and in endometriosis, which are both female reproductive tract diseases, especially in actually polycystic ovary syndrome, there's high levels of VEGF. So the blood vessels are extra leaky, even more leaky than during ovulation. So one of my thoughts maybe for future research was to actually try and treat polycystic ovary syndrome using liposomes to decrease the high levels of VEGF and to do more regular cycles and ovulation cycles for women that suffer from the syndrome.
Thank you very much, Maria. Our next talk is going to be about a comparison between species from primates to canines and to rabbits with nanoformulations. It would be presented by Pamela Schimmer and Michael Valic. I hope I pronounced it right.
Hi, I'm Pamela, and I'm going to be presenting on selecting animal species for the evaluation of nanomaterial toxicokinetics. The species in question are primates, canines, and rabbits based on the work of the Zhang lab at the Princess Margaret Cancer Center. When testing nanomaterials, you have to submit toxicokinetic studies in non-rodent species. Currently, there's more data for nanomaterials in canines and primates because they are more similar to humans physiologically and immunologically. There are many options of animals to use, and the one that you select is very important. In normal tox studies, either canines or primates are chosen as the non-rodent species in question. These studies are very expensive, costing up to $500,000. In nanomaterials research, both canines and primates are often tested together because there are concerns surrounding which model is optimal at mimicking human toxicokinetic reactions. This is very resource intensive, and we ask if there is a strategy to replace, reduce, and refine animal testing in nanomaterial toxicokinetics, possibly by using other species such as rabbits. We work with porphosomes, which are nanovesicles around 100 nanometers containing porphin lipid. They're very similar to pegylated liposomes, and we tested the toxicokinetic effects of infused porphosomes on rabbits, canines, and primates while maintaining a constant dose per weight across all the animals. We found that porphosome pharmacokinetics are very similar in rabbits and primates, but in dogs they have much variability between the test subjects. We also found that the porphosome concentration in tissues at the 24-hour time point were similar across the three species. Another thing we found was the safety of porphosome administration was similar in both rabbits and primates, yet infusion reactions were seen in canines which were not observed in the other two species. In conclusion, we found a 3R strategy to toxicokinetic studies in non-rodent species, specifically to our experiments with porphosomes. These include replace, reduce, and refine, where you can replace canines as being the default test model. You can reduce the number of primates used by doing the explorative experiments in rabbits and confirmatory experiments in primates. And we found that we can refine the number of animals used by combining data from all the species tested. This reduces the total number of animals needed. We have some key takeaways from our research that can be applied to nanomaterials research in general. One thing that we learned is not to overlook rabbits as a starting species. Since they weigh around the same as primates, they're a good model for primate PK. While studies should start with rabbits, they shouldn't just end there, as other species testing, such as primate testing, is important. Starting with rabbits can reduce the number of primates needed for the study, though. Another takeaway is to be cautious about resorting to canines as the default species, particularly for pegylated materials. And a final point to extend to nanomaterials research is the concept of using data from multiple species to reduce the number of total animals. With that in mind, thank you for listening, and a very special thank you to everyone in the Zhang lab.
There is a question from the audience actually, and it was kind of my question too, that did you investigate all your studies in rodents too? Because all labs start from rats and mice.
Yeah, so I don't know if you can hear me.
Yes.
Okay, so yes, of course, we always start with rodents, but when you're presenting tox studies, they always ask for a non-rodent species. So this would be like the next step going into more higher ranking, I guess, species just before human testing.
But how did that relate to your higher species, if you want to call them that? So how do the rodent studies correlate with?
So if the rodent studies are successful, then you move up, I think is what you're asking.
Okay.
I think Marcel Ali has a question.
Okay, I'm going to shoot this off. I'm wondering if you've done any of this work in consultation with Health Canada or the FDA. In my experience, when it comes to doing these formal toxicology studies, the work is actually guided by those individuals. And even though there may be some science to support an alternative, they often want to use a particular approach. And that would include a specific number of animals per arm, male and female animals, and invariably two-species toxicology.
Right. So I think there has been significant work with Health Canada. A good person to ask about this would be Michael Valic in the Zhang lab. He's been dealing more with that side of all of it as well. But yes, there are regulations that sometimes they do ask for that I guess you can't really get around.
Okay, thank you.
Thank you very much, Pamela.
Thank you so much.
The next speaker of the series is Ben Ouyang, and he will talk to us about dose threshold of nanoparticles, and he's from University of Toronto. Please.
Thanks, can you hear me?
Yes.
Great, I'm gonna share my screen and share the PowerPoint. All right. Hi, my name is Ben Ouyang. I'm from the Chan Lab from the University of Toronto, and today I'll tell you about the dose threshold for nanoparticle tumor delivery. So nanoparticles are designed to be delivered specifically to tumors, but the efficiency has been quite low and unchanging over the last 15 years. So there's a little bit of room to improve. The major barrier is the liver, and generally nanoparticles can deliver to tumors, but most of the nanoparticles seem to get stuck in the liver. And so at the Chan Lab, we hypothesized that the liver's ability to take up nanoparticles was finite. We asked the question, is there some dose where the liver would be overwhelmed? And beyond this dose, could we improve delivery to tumors? And so to answer this question, we vary the doses of many types of nanoparticles. And so for the purposes of this presentation, I'll just focus on gold. So we used 3 sizes of gold nanoparticles that were fully pegylated. And we used doses that span 5 orders of magnitude to really make sure we could find that threshold dose. And we injected them into mice with tumors, and we measured the biodistribution. And so we found that threshold. We saw a decreasing trend in liver accumulation as we increased the dose. There was an inflection point at around 1 trillion, which represents the threshold dose. And we'll explain the origins of this number in our publication that you can read later on. There was a mirrored increasing trend in tumor delivery as we increased the dose. And notably, I want to point out that at the highest dose, there was more in the tumor than the liver. And so higher doses beyond a threshold increased tumor delivery. Now, this inverse relationship between the liver and the tumor were connected by dose-dependent pharmacokinetics. At our lowest dose, what you'll see here is that the 50 billion, more than 80% was cleared out by 5 minutes. And at our highest dose, only 5% was cleared. And so this could also be described in terms of half-life, where our half-life increased from 2 minutes all the way up to 8 hours. And the only thing that we had done was just change the dose. And so how is this really connected? Well, since the liver took up less at higher doses because it was overwhelmed, more nanoparticles remained in the blood. And as more remained in the blood, more was able to deliver to the tumor. So then we asked, okay, well, can this be applied for therapy? So we used calyx as a therapeutic particle. It's a liposome filled with doxorubicin, as we've heard a lot about today. We hypothesized that if nanoparticle dose number mattered, then increasing just the number of nanoparticles should be able to improve the therapeutic efficacy independently of the drug dose. So we kept drug dose constant. And so we made a bunch of liposomes like calyx, but we didn't load them with any doxorubicin. So their only role was to overwhelm the liver. And so what we did was we had 4 treatment groups of mice, a sugar water control, empty liposomes only, calyx only, and combined empty liposomes with calyx in a co-injection. And we saw that while the empty liposomes over here, represented in gray, also with the control in gray, had no therapeutic effect on their own, and that calyx had an effect on its own. When you combine the empty liposomes with the calyx, we were able to receive an extra 60% reduction in the tumor volume, and also 30% increase in median survival. And so this really demonstrated to us that increasing nanoparticle doses beyond the threshold can improve the efficacy. And so in conclusion, there's a dose threshold that has to be breached to enable significant tumor delivery. And this is the Chan Lab. We hail from the University of Toronto's BME program, and these are the critical people that made this study such a joy to work on. And these are also my funding sources. And I'll take any questions now. In the meantime, I'll plug our paper that came out last month, and it has much more detailed explanations. And you can reach out to me or Warren on email or on Twitter. Thanks.
There's a question that is asking what other delivery strategies can be used to increase tumor specificity.
I mean, can we clarify this question? I think there are lots and lots of delivery strategies that nanoparticle engineers have tried over the last 30 years or so.
Okay, well, I would ask why was there an enhanced decrease in calyx plus emptied? That's what the person is asking. Is the best strategy to overwhelm the liver at the expense of better efficacy?
So there was an enhanced decrease in the calyx plus the empty liposomes because nanoparticle dose number matters. And so when we were able to increase the nanoparticle dose simply by adding the empty liposomes, that was able to increase the delivery to the tumor.
Okay. And somebody's asking, have you already published the data?
Yeah, you'll see on this slide, this is the paper that we have that describes our data. So you can look it up.
Okay. Yes.
May I quickly ask something? I'm just wondering if Marcel Bali has a comment about this, because I believe I've seen work from him or Peter Cullis like 30 years ago that had to do with dose of lipids and things.
I would say yes. I think the person that did this first is Terry Allen, and I don't know if she's still on at the meeting. So she really did talk about the importance of saturating the liver system. And there were certainly discussions about particle number from a liposome perspective, and that had to be done with dose. So in some cases, we vary the drug to lipid ratio just so that we can increase the particle number. That's what we described before. And I think Immunex Pharmaceuticals actually developed an IP position around taking drug-loaded liposomes with empty liposomes in order to achieve this particular effect. So this idea of number, I think, is really important. And then if you start thinking about it, because some of the other lore in the literature says that small particles are better than big particles, it really doesn't have to do with the particle size. It has to do with the particle number. And I don't believe we've actually published that, but if you inject the same number of particles using a 200 nanometer liposome, you get quite comparable pharmacokinetics to a 100 nanometer liposome if you control the particle number.
That's amazing. Yeah, that's actually what we verify in this study as well. So in the supplementary data of this paper, what you'll see is that depending on how you normalize your dose, if you pick surface area, or if you pick mass, or if you pick number, you get very different results and conclusions and interpretations of the data. And so if you normalize, for example, by surface area, what you'll see is that you start to get these size-dependent effects, where 15s get better delivery than 50s, get better delivery than 100s. But if you renormalize all of the data on this curve into nanoparticle number, they all just lie on the same line. So, yeah.
Yeah, so it really is an important observation, and I'm glad it's being revisited now, and I think it's something to think about, because sometimes we restrict ourselves to size on formulations, and I never understand why we do that. So,
And Dr. Valley, I wonder about your opinion on this, but right now, all of the clinical nanoparticles are really described in terms of the amount of drug that's given, right? So for example, Calyx is given at 50 mg per meter squared for a patient, but they never talk about the number of nanoparticles that they give. I wonder if it'd be interesting to describe doses in the clinic in terms of nanoparticle number as a supplement to the amount of drug that's also given.
I think that's being done preclinically, and the challenge here is that we're dealing with a system that's already been in place, that people define the dose based off of milligrams per kilogram. If you want to define it on the basis of particle numbers, all of a sudden you're having to change things. Like, in order to get the same dose, you're now changing the drug to lipid ratio for the formulations, and that can actually be quite challenging. Or you're diluting in empty liposomes, and that becomes a separate question from just a CMC perspective. That's difficult to do. But I think what really opens up here is that you can really start thinking about, why do we not use 200 nanometer liposomes? We could. We just need to think about the particle number that we're injecting.
Great, thanks so much.
Okay, I think in order to stay on time with the program, I have to move on to the next talk. Thank you, Ben, and thank you, MariaPoley, for the insight. Our next speaker is Richard Holland, and he is from Genevant Sciences in Vancouver, British Columbia. Please.
Thank you. Can you hear me okay?
Yes.
Good afternoon, and thank you to the organizers for the opportunity to give a short talk today. Genevant is a Vancouver-based company focused on nucleic acid delivery. Our portfolio includes a clinically validated and approved LNP platform and siRNA ligand conjugates for RNAi applications. Next slide, please. Endosomal escape is a principal bottleneck for nucleic acid delivery, which is addressed in LNP by inclusion of ionizable lipids. In contrast, siRNA conjugates have no endosomal release properties. Our RNAi 2.0 platform is a two-component system comprising an RNAi conjugate and a pH-responsive polymer with the same targeting ligand attached. Both components are administered subcutaneously. Following endocytosis, the polymer is protonated and destabilizes the endosome, delivering far greater quantities of the siRNA conjugate than if administered alone. Next slide, please. In this NHP experiment, we compared our standard GalNAc conjugate platform to a leading industry benchmark, which is currently in phase 3 clinical trials. Both test articles behaved similarly. When co-administered, the polymer on our GalNAc conjugate displayed a pronounced benefit in activity shown by the green trace, despite the dose being reduced 5-fold. RNAi still exhibits almost complete target knockdown 3 months after dosing. This clearly demonstrates how enabling endosomal release can be for siRNA conjugates. Next slide, please. We know that ASGPR is a unique case due to its extremely high levels of expression and localization on hepatocytes. But how about ligand receptor pairs in other cell types? Here, we have identified a series of ligands with binding affinity for the mannose receptor CD206 on macrophages. A labeled version of the best ligand is clearly bound and internalized by macrophages, but not in HepG2 cells, which lack the receptor. In vitro, knockdown is only observed if the mannose conjugates are applied in the presence of an endosomal release agent, highlighting the criticality of this step. Next slide, please. We have made similar progress targeting hepatic stellate cells. Again, we have identified a ligand that binds with high affinity for a receptor on the surface of HSCs. As with macrophages, we only observe activity when siRNA conjugates are applied in the presence of an endosomal release agent. We see no activity with negative controls. Next slide, please. In summary, we have designed a new RNAi conjugate platform with advantages over LNP. We have shown this platform works in NHP, considerably enhancing potency of GalNAc conjugates at 1/5 of the dose. We believe that our RNAi 2.0 platform can be used with other ligand receptor pairs, enabling delivery to other cell types and broadening the potential of RNAi therapeutics. Next slide, please. I'd like to acknowledge my colleagues listed here who have contributed to this ongoing project and to you for your attention. Thank you.
Thank you very much, Richard, for the talk. There are no questions. We will move to the next speaker of the series, Jayesh Kulkarni. He is from Center for Molecular Medicine and Therapeutics at University of British Columbia, and he'll talk about spontaneous solvent-free entrapment of siRNA with lipid nanoparticles.
Hi, my name is Jay Kulkarni, and I'm going to present some of my research on developing a solvent-free approach to encapsulate nucleic acids within LNP systems. Hi, my name is Jay Kulkarni, and I'm going to present some of my research on developing a solvent-free approach to encapsulate nucleic acids within LNP systems. LNPs are composed of 4 lipids: ionizable cationic lipids, phosphatidylcholine, cholesterol, and PEG lipids. And these systems are produced using rapid mixing techniques where the lipids are dissolved in an organic phase, the nucleic acid in an aqueous phase, combined through a continuous mixing device like a T-junction mixer, and the resulting suspension is the LNP. If this process is modified in such a way that the nucleic acid is removed and it's only pH 4 buffer that's combined with lipids in an organic phase, the morphology of the systems is dramatically different. It contains unilamellar vesicles. And that suggests that the difference in morphology between an empty LNP and those that contain nucleic acid is driven by the presence of siRNA. So we took a step back and we used these vesicles that form at pH 4 in a process called single-phase mixing. In single-phase mixing, the vesicles are combined with siRNA at pH 4. And it's called single-phase because they're all in an aqueous phase. This process is compared to the traditional 2-phase mixing, which is the organic solvent combined with an aqueous phase. And in both cases, we observe high levels of encapsulation. The particle sizes are essentially identical. And in our in vitro assay, where we look at the residual luciferase expression in a cell line that stably expresses luciferase, over the dose range tested, the 2 processes produce essentially the same particles. So what is the utility of this system? We chose to use it in rapid small-scale formulation screening approaches. So we produce preformed vesicles and we store them, and immediately before the cell treatments, we combine them with nucleic acid on the benchtop, and this process should dramatically reduce our nucleic acid requirements. We should be able to go down to as little as 10 nanogram per reaction. And so we tested this in a relevant setting, which is primary cortical neurons isolated from a transgenic GFP mouse, and we're knocking down GFP. What we observe is that the 3 processes, the 2-phase, single-phase, and benchtop approach, produce essentially the same knockdown over the dose range tested. So at this dose of 0.01 microgram per ml siRNA, we test a range of formulations. We start with KC2 DSPC, which is a very typical formulation, and we extend it to MF019, which is a proprietary ionizable lipid, and a range of helper lipids. So in summary, we were able to show that nucleic acid entrapment within an LNP does not require ethanol. Particle formation can be completely decoupled from entrapment, and this has implications for payloads such as protein. Preformed vesicles can entrap and deliver siRNA, and finally, that this benchtop approach can be used to screen through a number of LNP formulations and use very little material. And with that, I'd like to thank you for your attention, the organizers for the opportunity to present this research, and all the members of the groups involved in this. Thank you.
Thank you very much. There are a couple of questions. The first one asks, how long can performed vesicles be stored before they're used?
Yeah, that's a great question. So far, we've seen over time, of course, there will be a decrease in transfection potency, but we've seen it all the way out to about 6 months or so. Typically, lipids are particularly stable at pH 4, so you will see a decrease in activity over time.
Thank you. The next question says, is it possible to observe where the RNA is on the inside or the outside of the lipid nanoparticles?
Yeah, so it's definitely on the inside. I haven't shown this data in this presentation, of course, but using a combination of cryo-EM and dye exclusion assays, you can show that the RNA is internalized into the LNP system.
Thank you. And the last question asks, why would you do it this way? Aren't the final products the same?
So yes, absolutely. The products are very similar. So the key is really in is this formulation screening approach. You know, instead of actually having to produce a full formulation where you need a couple hundred micrograms of material for each round of formulation, you can decrease that to, you know, the nanogram scale. So that's one aspect of it. And then once you start to work with things like protein, like let's say the RNP, there's, you know, the role of organic solvents there is, it can be quite deleterious. So we're trying to avoid those.
Yes, and the last question asks that the difference between these two mixing methods, does it address particle morphology?
So I guess going back to some of the data that I showed on that slide, yeah, I mean, the resulting particles really are the same. And I'm happy to discuss in additional detail or send the submitted manuscript out, but it basically says that, you know, both methods are identical. So the resulting morphology is also the same.
Thank you very much, Jay. Moving on to the last but not least speaker of the series, Marta Overchuk. She is from Princess Margaret Cancer Center, University Health Network in Toronto, and she will talk to us about photodynamic priming. Please.
Hello everyone, my name is Marta Overchuk, and today I'm going to talk about how photodynamic priming can be used as a means of enhancing nanomedicine delivery and overcoming tumor dysplasia. One of the biggest challenges in cancer nanomedicine is that only a small portion of the systemically injected nanoparticles reaches the tumor. At the tumor microenvironment level, excessive extracellular matrix deposition creates a barrier against tumor nanomedicine accumulation and extravasation. In this study, we looked into the use of photodynamic priming, which harbors a photosensitizer and light, to break down this extracellular matrix barrier and hopefully facilitate nanoparticle extravasation. Photodynamic therapy, which utilizes photosensitizer and light to generate reactive oxygen species and induce cancer cell death and vascular destruction, is known for its ability to destroy tumors efficiently. However, its ability to modulate extracellular matrix remains largely unexplored. We developed a protocol in which an animal bearing dual subcutaneous prostate tumors was injected with a photosensitizer, after which one of the tumors was treated with laser and another tumor was left untreated. I'd like to note that we used about a half of a therapeutic light dose. That's why we refer to our priming as subtherapeutic. Next, animals were injected with gold nanoparticles, and 24 hours later, the biological effects of photodynamic priming were investigated. We found that subtherapeutic PDP did not induce tumor growth delay, cell death, or vascular destruction, as demonstrated by lack of caspase-3 staining or lack of decrease in CD31 staining. Next, we found that this priming, even though it's subtherapeutic, decreased tumor collagen content about 2 times, as demonstrated by the decrease in Masson's trichrome staining. Furthermore, we looked into the tumor ultrastructure, and we found that priming induced about a 2-time decrease in extracellular matrix density in the subendothelial zone. Therefore, it directly decreased the barrier that nanoparticles had to cross in order to extravasate. Next, we looked into the use of PDP to enhance the accumulation of copper-64 labeled liposomes. Using PET MRI imaging, we found that in PDP-treated tumor, accumulated nanoparticles a lot more effectively than the controls at all investigated time points. We also looked into the use of PDP to enhance the accumulation of clinical nanomedicine Doxil, and we found that it was effective at all Doxil doses explored. Finally, we found that photodynamic priming enhanced Doxil therapeutic efficacy, wherein a combination of PDP and 5 milligrams per kilogram of Doxil induced similar tumor growth delay to 15 milligrams per kilogram of Doxil on its own. Overall, we found that photodynamic priming has the potential to become a novel, minimally invasive tool to enhance the accumulation and efficacy of clinically proved nanomedicines in desmoplastic tumors.
Thank you very much. There is a question. How did you quantify the ECM density?
So can you hear me?
Yes.
We used two different methods. First, we used Messon's trichrom, so that was just used by quantitative histology analysis. And another method, we used transmission electron microscopy, and also we segmented regions of 1 micron around vessels, and then just based on the pixel density, essentially, we quantified the density of ECM around vessels.
Thank you. The next question asks, have you looked at immune activation due to photodynamic therapy exposure or adhesion molecule expression?
We used immunodeficient models, so we did not look into the immune activation.
And the last question says, was there any immune response at the unprimed tumor measured?
Yeah, that would be the same question. We used immunocompromised mice.
Thank you very much, and thank you to all the speakers for this session. I am not sure if we have a break or not. Urs?
I don't think we have time for a break. So it's a very strenuous day today. we keep going. But thanks a lot for all these short talks. It was great.